And we had that great lab activity where we're able to do transcription and translation on those boards. We learned that transcription, where we took DNA and we transcribed it into RNA. Now our focus was on messenger RNA. And we had a chance to work with those boards and we work with transfer RNA. And it's made the same way as any other RNA. There are genes that are devoted to transfer RNA and they just give rise to transfer RNA. They never translate into a protein because transfer RNA has its own function where it carries amino acids inside of protein production. And then we've also introduced ribosomal RNA, which is foundational RNA that makes the structure of the ribosome possible. And again, there are genes that are devoted to ribosomal RNA, and they are solely there to make RNA for the ribosome, and they turn into translated proteins. Any RNA is to be made by transcription. Messenger RNA is then translated. So when we talk about translation, this is only about messenger RNA, and it is translated into protein. So that's a brief review. Let's talk about that genetic code. How does translation take place? Now code, when they talk about code, they're usually talking about the genetic code. So they're talking about the DNA from which the RNA is transcribed. The genetic code is basically in reference, though, again, to genes, why they call it genetic. So those are discrete units on the DNA that are transcribed into RNA. When we talk about codons, we are specifically talking about messenger RNA. And we're focused on basically a 3-nucleotide sequence. And then when we talk about anticodons, we're actually talking about tRNA. So we can make some connections up here. Messenger RNA has codons, and tRNA has anticodons. And what is at the heart of all of this transcription translation is this complementary base pairing that occurs between the nucleotides. So if you have a piece of DNA, it looks something like this, AAA, APT, GGC, and then we found out, of course, that there has to be a complementary piece of DNA for replication nucleus, and for the complementary TTT, AAA, CCG. But one of these is ultimately translated into messenger RNA. Of course, we see the transcript into messenger RNA. And when we do that, we are actually picking one of these to be the strand that we are going to code from. So in this case, our example is going to be this top strand. And we're going to end up with making messenger RNA. So we'll go with A. Making messenger RNA. U, exactly. So we're going to have U, U, U. And then with this, we can match up the T. We know, of course, again, that A, because RNA is the same nucleotides except for it doesn't use that. And then G, G, C. This is our messenger RNA. I'm going to show you in a moment that tRNA is going to come in here. And we saw in lab that tRNA comes in here with this amino acids, and it actually matches up with these. We end up with this equivalent T or acid is it? So we can find that out by using this chart. So if we go for U, we go U, we start at the corner as phenylalanine. Probably the short form of that would be PAG. So phenylalanine would be the amino acid that's always attached to this tRNA. Every tRNA that has an anticodon of AAA gets phenylalanine on it, so it will match up here, and it will always result in phenylalanine. But it's fine. If we look at AAA, the anticodon for that, anticodon for that, so think about that. Make my little terrible flow leaves here, and we're going to figure out what that one is. So why don't you help me out? Remember, this is not what we read off that chart. We read the codon. Where the codon comes into play. So what's AAA going to be on that chart? Exactly. So right here, lysine goes A, up here to A, over here to A, and that lines us up with lysine. So lysine, what's going to happen next? And then I'm going to show you in a minute, we're going to end up linking these two amino acids together to start our polypeptide chain, and we're going to break one of these connections here, that one, and continue this process. So anticodons or whatever on the tRNA, the long-winded version. So why 3 bases? Why is the codon chart based on 3 bases? Well, there are a total of 20 amino acids that the cell has to be able to distinguish when it's trying to make proteins. And if you were to use just one base equals one amino acid, the problem is that there's only 4 different bases. There's A, there's U, there's G, there's C. That means you can only have language for 4 different amino acids. That's not going to work. We need up to 20. If you have a language where it's 2 nucleotides, how many different combinations can you get with an A, a G, a C, and a U if you're using 2 nucleotides? You figure that out by multiplying the number of different choices, 4, by the number of different choices, 4, gives you 16, 4 times 4 is 16. Still not enough for 20 amino acids. We have 20, but 16 is not enough. Why don't we go 3? The same idea, I go 4 choices, but now we're going to go 3 times. 4 times 4 is 16, 16 times 4 is 64 different possibilities. Now we have enough choices for 20 amino acids, and we have some additional redundancy in the code. And that's exactly what we see. We see some redundancy in this code. For example, here we see that every code that starts with CU, 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 CU gives us leucine. So that's kind of a protection for the cell. The cell has a mutation in the 3rd nucleotide. There's a change in the 3rd nucleotide. It doesn't matter as long as the first 2 nucleotides are made CU, you're always going to get leucine. So it doesn't really change the protein, it doesn't change the function. So the redundancy is a protective mechanism. The other thing that those additional codes give us are 3 stop codons. That's this additional code that we can place where the stop signal. So scientists figured this out and said, hey, 3 bases, they predicted it, then they figured out the code, and all the evidence supports it that it's a 3-base code that works for DNA. So that redundancy of code is really important. All right, so this is looking at the details of transcription. Here we have an enzyme that's kind of a transparent variable circle here. This is RNA polymerase. We've already introduced to DNA polymerase 1, DNA polymerase 3. RNA polymerase is kind of a do-it-all kind of enzyme. It comes in here, and you can see it's opened up the DNA double helix just like in DNA replication, but it's only for transcribing one side of the DNA structure. So we see G here, but A there, G, C, and machine is up here, 1 U down here. So here's an A, and there's a U. So it's basically putting those nucleotides together. There is an error in the image, sadly. So RNA polymerase is capable of both polymerase activity and helicase activity in one. It doesn't have the same issues with DNA replication. We have to pull the DNA completely apart in order to make 2 DNA molecules. it only has to open things up temporarily so they can actually start bringing in nucleotides to make the RNA. And once it's done with that region, it can close it back up again. So it opens things up, and then remember, polymerase is only going to add nucleotides together along that sugar phosphate backbone by creating covalent bonds there. And again, what drives all of this is that complementary base pairing, where A recognizes T on the DNA, and then C recognizes T, G recognizes C, and U recognizes A on the DNA. And where the mistake is that they've got the ends of these molecules all messed up. We're talking about the 4' to 5', 3', and this strand is kind of flipped. RNA has to be made off the 3' end because the 5' end. That should be 3' end. RNA polymerase is limited just like DNA polymerase. It can only add nucleotides to the 3' end. So RNA is constructed 5' to 3'. A lot of times when they are writing out genetic maps, they'll usually draw a big line here and then put a gene along the line. The way in this little box there, they'll put the gene of interest. For example, I might put a gene called beta-galactosidase in here. And then what they refer to as upstream to the gene, they'll put a region they identify called the promoter. This is basically a place where proteins can bind to the DNA and as a way to regulate the gene. So this is the gene here. We want to transcribe that gene to make an RNA from it. But the cell wants to have some control over that, wants to be able to regulate that production from that gene in order to, turn the lights off when you're not using it. You want to use up energy when you don't have to. So a promoter region will control the transcription. I often use a switch, like you turn the light switch off, but in reality it's more like a dimmer switch. You can kind of turn it down or you can turn it up. You can bring it down a little bit, you can bring it all the way. You can turn it all the way off too, for that matter. But they're usually what they call some leaky expression for most genes. In addition to that, somewhere along the end of the gene out here is going to be a terminator region, a place that basically says stop. So it's not going to be the stop codons that we were talking earlier. This is more about telling the RNA polymerase to stop. Stop codons are to tell the ribosome to stop making proteins. But a terminator region, this is interesting because it's still pretty active in science. There's a few hypotheses out there about how terminator regions are supposed to work. And they all have various supporters and promoters and evidence to support them. But they haven't really figured it out completely. The terminator region is basically still just telling RNA polymerase, hey, you're done making this copy. Time to stop off and not transcribe anymore. Keep going. Questions? Alright, so that's transcription. That's just making the RNA copy. RNA polymerase gets in there, opens up the DNA helix, makes the RNA copy of one of those strands, and now we're going to take that RNA and we're going to do something with it. We're going to go and make a protein. Oh, but before we do that, eukaryotic cells have to go through an additional process. If we were bacteria, we would just make the RNA. In fact, because the bacteria DNA is in the nucleus, the ribosomes and the cytoplasm would jump right on the RNA and start making proteins even before they're done making the RNA. That wastes time when it comes to bacteria. But we have a separate system. Our DNA is inside of a nucleus. This is a nuclear envelope here. And because it's isolated, there's an opportunity to do some processing with the RNA before we send it out into the cytoplasm to encounter all those ribosomes to make proteins, whether it be in the endoplasmic reticulum or through those free ribosomes. So RNA processing is what eukaryotic cells do before we can actually turn it into a protein. So it only happens in eukaryotic cells, at least so far that we've found. One of the first steps that's going to happen, there might be a little bit of a change in order between your notes as you print it out and what here. So let you know this is, I think, a better order. One of the first things we're going to do is actually put a cap and a poly tail on messenger RNA. And we want to put that on because basically they're protective ends for the RNA. There's all types of RNAs, enzymes break down RNA in the cell. And by putting the cap on there, we're actually able to protect 5' end. And poly A means basically take this messenger RNA here. And you just add a bunch of A's. You're going to add a bunch of adenoids. A bunch. Maybe like up to 30. And that protects us. So even if something does eat away at this tail, the important stuff is still intact. If something attacks that end, the important stuff is still intact. And the cap also acts as a trigger or serves as a signal to the cell, hey, this is where you want to look for the start codon for making a protein. So again, poly A tail protects 3' end. Cap protects the 5' end. RNA. And directs the ribosome. Along the strand of RNA, there are both what we call introns and exons. And one of the next steps we're going to do is actually we're going to bring in some spliceosomes. These are enzyme constructs or made of protein that you can recognize the introns and they flip them out. They splice together the exons. And when we're done with this, we end up with what we call mature messenger RNA. This is ready to go out into the cytoplasm and now be translated. So exons are really important. For a while, scientists were like, what are introns for? Wait a second, there's still some questions about that because it gets spliced out. But one thing we know is that introns sometimes serve as regulatory elements, short sequences of RNA that can regulate the activity of genes. We also know that introns give eukaryotic cells some flexibility, what they call alternative splicing. So they can actually vary where they remove the introns. It's not a hard order there. And by doing that, they can actually make slightly different versions, and sometimes completely different versions of a protein that can be very specific for different tissues in your body. And they'll actually look for certain versions of these proteins or enzymes in your blood and help diagnose disease, for example. So we recognize that even though we don't have as many genes as we once thought in the human genome, we also have more proteins than we thought because it's not really one, what they used to thought was one gene, one protein. So you can actually make multiple proteins out of a gene based on alternative splicing by how they splice exons together. In some cases, even splicing exons from one gene to exons of another gene to make an entirely different protein. So it's a wild world out there in biology, and there's still very much an active science trying to understand the proteomics of the cell, what kind of proteins are made from different genes. So RNA processing is an important part of eukaryotic cell protein production because the RNA has to be prepared for it and ultimately gives it some flexibility in what kind of proteins it can make. Translation. So again, reminder, this is taking messenger RNA and turning it into protein. So the previous image, this is sort of like the more realistic ribosomes. It took them a while to figure out the shape of the ribosomes, and it's a big big structure and took some small X-ray crystallography for us three-dimensional shape. But you can see the large subunit up here, the small subunit, they come together. You wrap around this messenger RNA, and they read that messenger RNA like a tape and tells them what kind of protein everything that we have that we've modeled in lab, so it gives you a leg up and hopefully understanding this material. And in lab, we got a simplified version of ribosomes, the way that I was taught ribosomes, a large subunit here, a small subunit down here, and there's three important sites in the ribosome. There's the E site, called the exit site, because when we get the tRNA in there, it's going to leave. We have the T site, what I would call the peptide site, because ultimately this is where the polypeptide ends up being still attached to the tRNA during the process. And then we have the A site, where the new amino acids are going to come into the ribosome during the process of translation. So we only get the E site, right? The exit. We call it spent. Does that make sense? Spent tRNA. tRNA is lost its amino acid. It's done with the process. And the P sites, tRNA with the polypeptide or peptide. Sounds good. And then we've got the A site or tRNA. Again, you see the big theme here with new amino acid. As we'll see. Keep that in mind. Get back on working with those boards. So a ribosome is ultimately a protein factory. It's where protein production takes place, and we're just going to get to the process of how it happens. You can see in this image also that this tRNA has this anticodon, and there'll be a complementary base pairing between it and the messenger RNA, CG, ADU. It's all RNA, so there's no binding. That label also shows anticodon and codon too. So I always get ahead of myself. That's okay. Again, notice that anticodon on tRNA. Even here, complementary base pairing is critical to this whole process. It was important in DNA replication, it's critical in transcription, and once again in translation, we need it. And there is a tRNA for all 20 amino acids, and the cell keeps, a healthy cell keeps a ready supply of all tRNA with those amino acids available so that protein synthesis never stops. If you stop protein synthesis in the cell for too long, it dies. That's how some antibiotics work against bacteria. It stops protein synthesis in the bacteria die, and you get better. There's some bacteria that make toxins that stop protein synthesis in our cells that makes us sick. So it's a critical feature of cell function is making proteins. Transfer amino acids to, so what do tRNAs do? tRNAs transfer the amino acids to the site of protein production. We talked about that last time we listed all the RNAs. A little redundancy here is also our friend. And this little table here is quite pretty hard to see from back there. It's showing us messenger RNA codons, the anticodons, and the amino acids that associated which we get off that table. So let's talk about translation process in more of a step-like pattern here. Our initiation involves the small subunit binding to the messenger RNA. That's what we're seeing up here on this image here. The small ribosomal subunit is grabbing onto it along with what's called the initiator tRNA. And recognizing that start codon is so important. And bringing with it methionine. Once that's on there, it's basically a trigger for the large subunit to come in and lock on. Then the methionine tRNA gives it a little place to hop on and travel through the messenger. So now we have one big happy family here. Initiation is done. So just like I sort of drew on the board here, or a backwards fashion, this takes it sort of midway in the process. We can see methionine is already right here, so they're pretty far down the process. But the idea is that we start off here with a tRNA that has the peptide or an amino acid. Here comes in a new tRNA, the anticodon lock matches up with the codon, and as we learned in lab, we don't transfer the ASP, this aspartic acid, to the valine. We need to transfer the entire polypeptide from this tRNA to the new tRNA that's arrived. Form that covalent bond, we form the peptide bond. There's the peptide bond. Now the whole thing goes to this tRNA, and now the ribosome can move downstream, and the tRNA that no longer has an amino acid is on the E site, so it exits. When it exits, the cell says, oh, you don't have an amino acid. It brings in an enzyme, and it adds an amino acid back to it, which we learned that was valine for this particular tRNA. And then this repeats. We bring in another tRNA. Now UGG is matching up with ACC. We're bringing in threonine, and we're going to move that aspartic acid for a polypeptide bond there. And this is going to shift again. We're going to kick this one out. This whole thing will be touched. So that's elongation. That's just repeating that process over and over again, adding amino acids to the growing protein, forming those peptide bonds. And then termination is finding that UGA or UAA or UAG, proteins like that, and that's the stop codon. That's when the release factor comes in. You can't form a peptide bond with it. That's the signal for the ribosome to come apart to break the last bond of that tRNA and read the now mature protein, or potentially further processing in the Golgi apparatus, and of course might turn into a glycoprotein or be folded in a certain way, and then be distributed based on where it needs to go. So those are the stop codons again. You should know those. And it's a signal for the ribosome to say, let go. Let's change it up. Why don't you pull out a piece of paper and we'll put down the activity. Hey, yeah, now remember, you don't need to have a device to do this. You can just write down the answers on your activity paper. Of course, I encourage you to go and use QR code, or if not, you can enter in this. So it has a number of ways of controlling the expression of its genes. One is at the level of pre-transcriptional control. So this is in reference to the variation that we see in chromosome DNA availability. Turns out that our chromosomes, this has to do with also with how different tissues are formed from different, from the same chromosomes. We all have 46 chromosomes in all our cells, and yet blood cells are very different from muscle cells, bone cells are different from blood cells, and neurons are different from all of that. So how do we get so many different types of cells? Well, we're using 46 chromosomes. And it has to do with the fact that some of these cells have turned some of their genes permanently off because they are genes that are not for that particular tissue type. And they do that by condensing the DNA so tightly that it's not even available for transcription. One of the terms that I won't test you on this that they use for this is called heterochromatin. And it's really tightly packed DNA that's just not available. There's what's called epigenetic control, as well as methylation and ubiquitization of DNA. So it's just available. This is kind of what they're showing up here up in this corner. We've got the DNA wrapped in histone like we normally find, but it's wrapped really tightly around those spools that it's not even available for transcription. So we refer to that as pre-transcriptional control, even before we get to this level. So if the DNA is too condensed, that's the cell control at a pre-transcription level. It's usually turning the gene off permanently. Transcriptional gene expression control is probably the most common type of control of genes, because this is kind of one of the best places to do it. If you want to have a gene still available, we're actually kind of turning it on and off as needed, then doing it at the transcriptional level makes a lot of sense, because if you never make the RNA, you never make the protein, you never waste the energy on the RNA making, you never waste the energy on the protein making. At these other levels, you have to spend certain energy. So if you do it after transcriptional level, then you made the RNA and it costs the cell energy. It's not the most efficient way to do it, but sometimes it's the best way to do it given the circumstances. We'll talk a little bit about when that might be good. But controlling here in terms of transcription, done with a lot of different transcriptional factors. Either there's proteins that have to do with this whole promoter region I was talking about earlier. Some proteins will bind this, will increase the expression of the gene. Other types will bind it and will inhibit it so that RNA polymers won't come on here and transcribe the gene. Some very elegant ways of doing this. The most common, one of the most well-known or understood ways is it involves this particular gene that I wrote up here called beta-galactosidase that bacterial cells use to process lactose. Lactose is an uncommon disaccharide sugar in the environment for bacteria. And so if there's no lactose in the environment, why make all of the gene products necessary to process lactose? It's a waste of energy. So if there's no lactose, there's an actual protein that binds to the promoter and blocks the transcription of this gene. When lactose appears, the lactose binds to the protein and causes it to release the promoter. And now the RNA polymerase comes in here and starts making beta-galactosidase, which breaks down the lactose and it turns it into glucose and lactose, which is something the cell can then use for energy. So it only turns it on when there's lactose present, and lactose is what activates it. I mean, really an elegant system. When lactose is gone, then the protein comes back on here, but no more lactose bound to it, it turns it off. The cell stops making enzymes to break down lactose and saves energy when there's no lactose around. A lot of transcriptional control occurs that way. Post-transcription basically is referring to after we've made the RNA. And this is typically at the level of RNA processing. It's probably the least common of the types of gene control, but it's possible to make it so the RNA is not processed, and so you don't get mature RNA to actually translate into protein. So I said post-transcriptional control. But right here at this point, the RNA never gets processed, never gets put in protein, therefore it's never available for making protein. It's a little bit wasteful because you spent energy on the RNA. So why would this be a good level of control for the cell? When might it want to use this? It's a factor of time that's important here. It takes time to transcribe RNA, so if you have a bunch of RNA that's pre-transcribed, it's faster, so if you want to turn the gene on quickly, to have some of this around, then you just have to process it, send it out, make proteins. If you start at this level, then you have to make the RNA first, process it, so it just saves a step. Sometimes cells may be post-transcriptional control to save a step to make it so they can make proteins a little bit faster. And that's really kind of the theme for a lot of the rest of these. Translational control is achieved by that RNA that we talked about last time called sRNA, short interfering RNA or microRNA. So basically, it gets in there and it stops translation. It stops the ribosome from making the protein. So in many ways, it's very similar to post-transcriptional, but it's at the level where we have a mature RNA. So stops translation of mature RNA. And it uses sRNA to do this. So a lot of times here's your messenger RNA. And we'll put a bunch of U's here. A's, G's, C. And here comes a little sRNA. And it goes ahead. And actually, let's change this. Let's make this right. So that's the start code, right? And here comes a little sRNA that has the following sequence. U, A, C. It comes in there and it latches on this, forms some hydrogen bond between them. It doesn't go off. Now the ribosome can't bring in the initiator RNA. Like, I can't put the methionine in there. There's something in the way. That's what sRNA does. It blocks the translation. You can actually also just basically target the mRNA for destruction too. But again, if you have the mature RNA already lying around, if you have a process to remove this later on, then you can go ahead and translate this more quickly than have to make the RNA, process the RNA, before you can get the protein. So again, it's a step-saving method, although it does require energy investiture. And of course, messenger RNA doesn't last forever. It doesn't store well. It doesn't break down. So there's that problem too. Post-translational. So how can we control genes post-translation? That means we actually have the protein now. How do we manage to control that? This is done at basically the level of protein, where we actually regulate what the protein does. Imagine, proteins are complex. Alright, so say you have a protein, let's draw some sort of random shape. I'm going to put a little shape in here. And then I'm going to put this big site here. This is what they often call the active site. So there's always basically a place where the protein interacts with its substrate. And we'll bring in the substrate in a different color. Substrate is what the protein or enzyme will act upon. So here's the substrate. And this could be all kinds of things. Say, for example, this could be lactose. That's a sugar that the enzyme will break down into a disaccharide monosaccharide to make sugar available. But what happens when we don't have lactose around? Maybe its substrate is an amino acid, or the parts of amino acid to make an amino acid. When there's plenty of this amino acid around, sometimes with all this amino acid out here, when you attach around, one of them goes over here and binds into the protein. So it has plenty of amino acid. When it binds to that, it closes this site down, it pinches this shut, so that it can't accept any more substrate. If the cell has plenty of this around, it doesn't want to use the protein up anymore. Then it starts using these amino acids, and eventually uses this one, pulls that out, this opens the site up again, and starts making amino acids. Sometimes cells will modify these proteins by putting phosphate group on them. This is the active form of the protein. And then later on, when it doesn't want to work anymore, it takes the phosphate off. A couple of enzymes called kinase and phosphatase. So it has a mechanism by which you can turn protein. Think about the little on-off switch on the protein, like a computer. You turn it on when you want to work with protein, you turn it off when you don't want to. Cells have mechanisms to do that. Some of them are quite elegant, what they call feedback inhibition. The earlier example where I showed the substrate coming back and inhibiting the protein, that's feedback inhibition. It's making the protein not feeding back and stopping its activity. So that's a very common mechanism for protein regulation. Questions? Thoughts? All right. Let's talk a little bit about DNA technology. It's been an entire semester talking about this, but we're going to do it in just 15 minutes or so, maybe less. So the book goes into quite elaborate detail about all this. I want to keep it pretty simple for you so we can explain some assessment questions with the best that makes sense. So what is polymerase chain reaction all about? It's also called PCR. It always involves these machines called thermocyclers. You program them to go 95 degrees to denature DNA, 55 to allow the primer come in and find this target. These primers are designed specifically for a specific segment of DNA or a type of DNA. They usually design these primers so it's a really dark form to it. You have to get to a certain temperature so that they actually form hydrogen bonds. Remember, if you heat up DNA, it actually becomes a target right away. Then you bring it down to 72 degrees, and it uses an enzyme, uses the DNA polymerase, and the DNA polymerase has been isolated from a bacteria that they got from Yellowstone's National Park, that actually grows in hot springs. And the enzymes of the bacteria are designed to work at a very hot temperature, almost boiling point. And so it doesn't denature when you bring it to 95 degrees Celsius. At 70 degrees Celsius, growth works at its best. So it makes a DNA copy using that primer. Our DNA polymerase needs a primer. It's usually an RNA primer, but for this reaction, you use a DNA primer. It works just fine. And then you recycle. You do this again and again and again. You heat it back up to 95, cause all the DNA to come apart. You cool it down, allow the primer to find the DNA. Bring it back up, allow the DNA polymerase to make copies of DNA, and you repeat. You do this like 30 or 35 times. And at the end, you end up with lots and lots and lots of copies of DNA. And remember, with our experiment, we were isolating DNA from strawberries and bananas. The reason we could see the DNA is because we had so much of it. We had broken open millions of cells of strawberry and bananas to free their DNA and became this globular mass of DNA from all those cells. Then you can see it. It's the same idea here. We make lots of copies of DNA so we can see it. Say right now there's a little cell inside of your lung. Let's not make it perfect. Let's say someone goes to the hospital, they're sick, and they want to know, does this person have tuberculosis? Maybe there's only a few cells of tuberculosis, mycobacterium tuberculosis, in their lungs causing disease. Now there's two ways you can do this. You can take a culture and you can try to grow the bacteria. Tuberculosis is famous for growing very slowly, so it can actually take 6 weeks, months to get this broken up and strained. But meanwhile, this person's sick and you want to diagnose them and treat them as soon as possible to have a chance to save their life. With PCR, you actually will detect a few copies of tuberculosis bacilli bacteria with this technique in a day. By the end of the day, you have a diagnosis and that person treatment. What's more, there's even advanced PCR technique that will tell you if the bacteria is actually resistant to certain antibiotics, so you can actually get them the right antibiotics too. So it's an extremely potent technique that allows you to take a few, which you can't see, and make a lot of copies of that you can see, although it requires special equipment, but you still can do it. And you can diagnose. You can also detect whether there's a few copies of DNA in a crime scene. You think someone is caught committing a crime, you make specific primers for that particular person's DNA, like a fingerprint. They will identify what their DNA was at a crime scene. So it's used in forensic medicine too. It takes a few copies and makes lots of them. So it's a very powerful technique. And that's what it's made for, making lots of copies of DNA. Sequencing. In order to make those primers, you have to know what the sequence of the target is in the first place. Those primers, example, could have been specific for tuberculosis and no other bacteria, so they don't recognize E. coli or Streptococcus pneumonia or any other bacteria, only mycobacterium tuberculosis. But you can only do that if you know the sequence of the DNA you're trying to target. How do you figure that out? That's where sequencing comes into play. Nowadays, a lot of sequencing is automated. A lot of it use polymerase chain reaction, micro sequencing reactions used it. And it involves basically interrupting the process of DNA replication using nucleotides that have specific fluorescent markers on them. so that the fluorescent marker on the adenine is green, fluorescent marker on the cytosine is blue. You don't have to do fluorescent marker with an automated, you have to use fluorescent marker. You can do what's called sequence shells. You spread out all the different fragments of DNA, and then you actually read those fragments like you're reading what's A, B, T, G, C, C. It takes longer. This automated sequence is pretty cool. The fluorescent marker is the computer reads the fluorescent signal by shining a UV light on there. And so it sees green, so that's an A, and it sees blue, it says, oh, that's a C. And so it literally determines the sequence of any given piece of DNA. So it becomes so automated and so efficient that now we know the entire genome of humans and a lot of other organisms besides. It's becoming more and more efficient as time goes by. So if you want to know the actual sequence of nucleotides for a given piece of DNA, sequencing is how you do it. Cloning. Yes, you can clone organisms. They clone the sheep, but that's not what we're talking about here. Here we're talking about cloning genes, making copies of genes. So it sounds a lot like polymerase chain reaction. Polymerase chain reaction mostly focuses on making copies of a particular segment of DNA, not the entire gene. The entire gene is pretty much out of its purview. But taking a piece of DNA, so we'll use a specific example because it's a great example. Here they're taking the gene for insulin, and then they're using a bacteria. And bacteria have these pieces of DNA called plasmids. They replicate independently of the chromosome. The bacteria make lots of copies of them. We've actually isolated them and designed them, so we call them designer plasmids, so they do specific things for us. And you go ahead and you cut it with an enzyme called restriction enzyme, and then you rejoin it with this insulin gene using our good friend ligase. We talked about the DNA replication. It's really good at linking together pieces of DNA. And now you've got a piece of DNA that's part bacterial DNA, part human DNA, and it has the insulin gene in it. Then you go ahead and you put it back inside the bacteria, and the bacteria does what bacteria do. You give it food, it grows, and every time it grows, it makes a copy of the chromosome. And then from that chromosome, it reads, or from that plasmid DNA, it reads that piece of insulin and it makes insulin. You isolate the insulin, now you have pure insulin to give someone and save their life from diabetes. They did this and they found out that the insulin gene had all those darn introns in there, and they said, oh, we can't do that because bacteria are prokaryotes, they don't know what to do with introns. So they went ahead and isolated the messenger RNA that was mature that had the introns removed, and then used an enzyme called reverse transcriptase to turn the RNA back into DNA. Then they put that DNA inside of one of these, they put it inside of a bacteria, and isolated a lot of insulin. Now you have really pure insulin. They've done this for other compounds. So cloning genes, making a copy of genes, and allowing the organism to produce the actual product from that gene, whether it be insulin or growth hormone or some other protein that's useful to us in society. And this newest technology is the one thing on this list I haven't done. Editing genes using what's called CRISPR technology. This is actually a particular type of enzyme called Cas9. And it's interesting because it uses a complementary piece of target RNA that actually recognizes, they call the guide RNA, and it will find in the genome that particular specific piece of DNA as a complementary base pairing. There's nothing else like it. It has to be unique. In fact, that's one of the challenges with this technology, is making sure you have a sequence that only recognizes the target DNA, because if you recognize something that's not the target, this is with putting mutations in the DNA, so you can actually mess things up. And they're using this to fix errors in DNA, to do gene therapy, to try to cure people of disease, of genetic disease, so they need to get it right. If they get mutations, they do modify system, they're going to mess people up. Showing that they create a small deletion, or what they call a point mutation, where you change one nucleotide, or even encourage it to swap out an entire piece of DNA for another. This way you can really fix a gene. This is potentially a way to fix something like sickle cell anemia, which is only a single nucleotide change. And give someone the correct gene so that they make normal hemoglobin, so that their red blood cells don't form that sickle shape and it makes them so ill. So we can fix genes also, although that's still a developing technology. So using all of these different techniques and tools, and that's just a small, very, very small sample of them. We've done a lot of genetically modified organisms. Genetically modified bacteria, as I mentioned, we've modified bacteria to produce insulin. We've modified bacteria, actually, yeast cells, to produce the hepatitis B vaccine, a very effective one. They identified the proteins in hepatitis B that would give the best immune response. They isolated that protein, they cloned it into a gene, and then they would have produced lots of it and use it for vaccination. It's been very, very effective. Human growth hormone, we've also cloned that gene and put it inside of bacteria. So they get bacteria. This is basically showing a bunch of flasks of microbes growing, and inside there, whatever they're producing, the insulin, the growth hormone, I'm not sure why it's called names like microbes there like that. That's chunk change. That's a very small product. This is how they do it industrial. They make large fermentation vats and they grow tons and tons of it to get a lot of product to treat people with. They modified plants. They make crops that you can spray Roundup, which is a pest, an herbicide, and it doesn't affect them. So you can spray your crops and kill all the weeds, but the crops survive. However, there are a lot of people who are not particularly happy about Roundup being sprayed upon their potential food. So it's got some kickback. We've made drought-resistant crops. A lot of food, a lot of crops now are genetically modified. They've been modified so that apples don't brown as early or as quickly. Because people don't like that for some reason. They find it visually unappealing, even though it doesn't really affect the taste that much. A little bit. They've even modified animals, faster growing salmon, so we get more salmon. You go to the store and you buy something that's not wild caught salmon, there's a good chance you're growing salmon that's been modified that way to grow faster and give us more salmon. They've modified male mosquitoes so they are sterile and they release them. They'll basically make thousands of these. They'll release them in the environment. The females will make them thinking they're normal, but they're not. And they don't have any with sterile eggs, and so they can't produce any babies, and that's cost production and transmission of malaria because there's not many mosquitoes. Actually hurts the population quite effectively. Literally thousands and thousands of human lives, I mean kids in particular, lives have been saved through this technique and limiting the spread of malaria. They even make goats that produce human growth hormone, so against the bacteria that you can make it from goats. You know, turned to treat human dwarfism and also as a potential therapy for serious injury. Growth hormone is pretty effective. We've also created mouse models, so that basically mice have very similar genomes to us. You can actually knock out genes, and then you can see what the effect it has on the mouse and try to get some idea how to treat humans based on the effects of that gene. So they developed a mouse model for breast cancer, they knocked out the gene for aromatase, which produces the estrogen, and they looked at what effect that had on breast cancer. That's one example, one of many examples. There are tons and tons of genetically modified mice out there for, say, all types of different genetic diseases. They modified pigs so that their hearts are acceptable as transplants. So our immune system tends to reject anything that it sees as foreign. That means even another human's heart can be rejected. So that they try to modify pig hearts so that it doesn't happen. We call these xenotransplant because they can transplant from other animals. They actually did a pig heart transplant not too long ago. The person did not survive for very long, but they lived for a little bit longer. The idea is to get it to a point where they can have a pig heart long enough to be a totally transplant is available, or maybe eventually sometime get it so they can just be accepted. Pigs are about hearts about the same size as us if they're I'm not going to go into gene therapy. I didn't for Thursday, so I'm not going to do it here. We're going to move on to evolution. This will not be on the test. Hopefully I have a rule on not having somebody to be analog presenting, just because I don't know. You're welcome to have it on the tabletop. So we're going to go into human evolution. It's really kind of everything evolution, and gosh, this is such a big topic. So we'll start very generically and talk a little bit about different processes and evidence for evolution, but we won't get into the details of macro and microevolution, which is kind of a sad thing. But we're going to start really at the very beginning here with the origin of life. So planet Earth's been around here for a little over 4 million years, and we believe life has been on this planet for anywhere from 3.5 to maybe close to 4 million years. Certainly some evidence for it being around, so I think 3.8 billion is the last one they pushed it back. And so one of the things, scientists basically just don't cringe at anything there. They want to find natural explanations for everything that happens that we can study, because we're always seeking knowledge, we're always curious, and it's just something that interests us. So the question is, how did life get started? And just thinking about doing thought experiments, people say, well, it makes sense to start small and build up. Just kind of the way we organize things at the very beginning of class. We talk about atoms and subatomic particles, and we put those together as molecules, and put those as macromolecules. And the same idea here. You start off with very small organic molecules. It has to be organic. Ultimately, you don't get different things. The life is made of organic material. Start with small molecules, and then become macromolecules. And you can see here their little chart for you. Early Earth, inorganic chemicals, energy capture, abiotic synthesis, small organic molecules, stage 1. Stage 1, small organic molecules. Then we start linking together through polymerization. We get larger polymers, what we call macromolecules. That's what a polymer is. So now we got bigger molecules where we actually get polypeptides, and maybe some nucleic acids, carbohydrates. And then we get our first protocell. And when they mean protocell, they mean before the cell. So the idea was that at this stage already organic compound polymers were linking together to make cell-like structures that didn't last. They would form and then they would die, they'd break apart without reproduction, without copying of DNA or anything like that. And they had some metabolic activity, they believed, but they did not have conquered the ability to actually replicate. And that's what you need. Sometimes they're called protobionts, is another name for these protocells. So the first cells have the capacity to actually copy DNA, RNA, and so arguing about what might have been the first nucleotide, but you have to have the ability to replicate those molecules, and there has to be some sort of impact upon them via natural selection, where the environment is going to select the cells that have the best ability to replicate themselves and their genes. Richard Dawkins wrote a famous book called The Selfish Gene, because it really kind of comes down to genes wanting to make copies of themselves and doing it the best way they can. Those that could do that the best were the ones that survived and made more copies of themselves, so that particular efficiency kind of got transmitted in later generations, and you got more and more complex and sophisticated life. But you have to get to the level of living cells for this process to be. You have to at least get to the level of self-replicating molecules that have some ability to respond to natural factors. So they can start this process of selection to select those that are the best to do it. And eventually you improve to the point where you get something that's us. So some of the early experiments, some of the ideas that supported this train or pattern was based on what we understood about early Earth. And that's a science in and of itself, because how can you tell Earth was like 4 billion years ago, 3.5 billion years ago? There's some clues, there's definitely data, this is not guesswork. There's evidence, people looked at ice cores, fossil records, and patterns like that. Geographical deposits of different minerals, all these have given us clues of what's happened on the planet in the past. And we believe, based on those clues, that the early atmosphere was mostly water vapor, some carbon dioxide and nitrogen, but little to no free oxygen. You would not have survived on Earth. Oxygen was a toxic chemical that reacted very quickly with other chemicals that were moved. Over time, Earth started off molten, like all planets, basically aggregate of a bunch of solid mass that was very, very hot to begin with. But eventually it cooled down, and as it cooled, that water vapor started to fall out of the sky. Heat the water up, turns into steam, you cool it down, it condenses and falls as rain. Oceans started to form in response to that. There was still plenty of energy. There's still lots of volcanoes. There are volcanoes even to this day that are active, but they're really remnants volcanoes, because there used to be a lot more when the Earth was hotter and younger. A lot of lightning, there's still a lot of lightning today, but there was even more then because the atmosphere was more violent. There was a lot more energy feeding inside. Volcanoes can cause light because there's so much energy in them. So that would be one form of energy to feed into that. A lot more radioactive isotopes. Radioactive isotopes decay over time. They become non-radioactive. So at the beginning, with all this energy, there was a lot of radioactivity, but over time, it's diminished. There's still radioactive isotopes to this day. There's a lot more back there. So again, it's a form of energy. More energy. Electrons flying all over the place, photons, neutrons. And even ultraviolet energy. There was no ozone layer. Ozone is a form of oxygen. So if you have no oxygen, you can't have ozone. And we are protected from the UV radiation from the sun by the ozone layer of this planet. There wasn't one back then. So all this UV radiation is hitting planet Earth, which also causes mutation, but also a form of energy. Stanley Miller was an early chemist who was looking for a doctoral thesis, and he wasn't really interested in a lot of things he kept reading about. Then he stumbled upon this idea of how did life originate, and he was fascinated. He wanted to try this experiment where he said, well, let's take what we know about early Earth's atmosphere, let's throw some things, some energy at it, and let's see what we come up with. So this is sort of the schematic of his setup. He had a boiler here. He's heating up some water to give us some water vapor. He's got some gases in here that we found in early Earth: methane, ammonia, hydrogen gas, and of course the water vapor. And then he's applying some electricity. He's got an electrical spark. He allows that to condense, as what would happen on planet Earth. It would fall in the rain. Liquids are collected, and then at the end, he looked down here, going to be fine. He found that he had gained some small organic molecules. Something that people didn't think that could happen spontaneously. But it turns out it happens pretty easily. We've actually, at this point now, we've found them on meteors and comets. So there's some debate about whether even comets and meteors may have seeded the planet with some more complex organic compounds, or maybe even life on another planet. But although that just really pushes the question back. So wonderful how life originated. If it happened on another planet, what happened there? Another type of thing that I like to bring up, another phenomenon they talk about are thermal vents deep in the ocean. Those areas are under such pressure that even water at boiling temperature doesn't boil. At 100 degrees Celsius, if your pressure is too high, water will not boil. So you actually get water even hotter than boiling point. And water. So there's a lot of energy there, plus the thermal vent is actually a vent for a volcano, an underwater volcano. So there's all of that, there's all the chemicals coming out of that volcanic activity, which using a rich stream of different atom types to form organic compounds. Stanley couldn't do this. He could not recreate this high pressure environment under the ocean. The problem with that high pressure environment is that it's not exposed to sun energy whatsoever. But still, there are proponents that say that probably the early life probably happened in darkness during these soil events, because others have worked with pressure, looked at what type of pressure got on forming complex organic compounds. But before I get into that, I'd like to add to our activity sheet, and I'd like to address these three statements. Compare transcription simulation, describe the function of two enzymes used in DNA replication, and describe one benefit of genetically modified organisms. Often, which macromolecule first? A lot of proponents for RNA first because RNA has both the capacity to replicate, so the whole complementary base pairing thing working for it, and it's been shown to have enzymatic properties. And part of the way the ribosomes work has to do with the enzymatic properties of RNA. Well, that's a lot of things going for it. Sidney Fox was a big proponent of protein first. He felt that proteins had the capacity to form cell-like structures, and what he couldn't show those, how they could replicate. And it's still really an issue. He did do a series of experiments, though, where he put some of the precursors that they were able to generate in the earlier Miller experiments, put them under real high pressure, and he built what are called microspheres. And he didn't build them; they just formed under these pressure conditions, and they had the ability to replicate to a certain degree. So he thought maybe this was a possibility, but still, no cell, wasn't nothing living. He passed away in 1998, back. So as I mentioned, there's, in terms of on the way to life, protocells is one of those steps, macromolecules is one of them, and RNA is still a big contender in that regard. There are viruses that only use RNA for their nucleic acid. They replicate, they don't have metabolism, so that's all they're missing, metabolism, and they could potentially begin to evolve and produce the first life. So some of the early protocells were probably what they call heterotrophic. Heterotrophs basically feed off of chemicals. Or they were doing photosynthesis, they were doing sort of oxidative reactions, at least not with oxygen, but they were, there's lots of chemicals around in this early primordial soup. So there was the potential for it to use these chemicals to generate energy. And so that's what I think probably was going on. They're definitely anaerobic because anaerobic means no oxygen. Literally anaerobic. Without oxygen. Aerobic means oxygen and means without. The first crude cells, if RNA was the first molecules, then somewhere along the road did RNA make DNA? Was DNA just a mutated form of RNA? I don't think we'll ever know. Unless we find another kind of earlier life that shows it's out there. Maybe that might be different than happened here. Did proteins make the first DNA? How did the first DNA replicate? How did the first cells divide? These are all questions that have yet to be answered. So we still don't know how to replicate the process of going from macromolecules to actually the first true living cells. There has been the construction of an artificial cell where basically someone put together all the bits and pieces, knowing what a cell is supposed to look like, but it's clearly not the same as what happened in this anaerobic world that was pretty much pretty alien to what we understand. So what we've been able to observe, though, is that life on the planet has changed. It became very apparent to early scientists right off the bat because fossils had already been discovered. Probably the earliest cultures found fossils, and that's where the mythology of things like dragons came around because they saw these giant bones from dinosaurs. They imagined some sort of monster that was responsible for that. And then as science became a little bit more sophisticated, they started looking at other fossils, and they saw that some of these fossils were very similar to living organisms that were still on this planet, and started coming to the idea that maybe the organisms that exist now evolved from organisms that existed in the past. That was not a unique idea. Lots of people pretty much understood that that had happened, but there was still a lot of debate about how did it happen. They didn't know about genes and DNA and RNA and all the things that we know now. They had much more limited information. They knew things about populations and how populations grew. We knew about the environment, how everything that is produced early in life doesn't necessarily survive. So flies have millions and millions of larvae, but only enough survive to replace fly populations. Fly populations grow, and then they decline. They grow and decline. Very tenuous existence of flies are always around. There's enough to reproduce, but they don't entirely cover the planet. So some of them die. Not everything that's produced lives. And that's what everyone started to realize. And even Thomas Malthus, an early mathematician, economist, basically talked about how even humans are all competing for enough resources to produce family, have children, and to carry on the next generation. And he started talking about ultimately the planet's a limited sphere. It's a limited space. How many people can you fit on it? And what decides who survives and who doesn't survive? And what features do they have that makes them more capable of surviving and getting enough resources to reproduce? And all of these are going around, bouncing around people's heads, and people are reading what other people have written about it. What we do know about the earliest life, though, is that the first cells were prokaryotes. They did not have nuclei. They were not aerobic. They were anaerobic. In fact, when oxygen first came onto the planet through photosynthesis, a lot of things died because oxygen was toxic to them. And to this day, there are organisms that are bacteria that you put them in the presence of oxygen, they die. And they're anaerobic. They're very strict anaerobes. They are descended from ancient organisms from the past that lived in a world where there was no oxygen. They've adapted to a world where there is oxygen. Eukaryotes came along because basically eukaryotes, size matters. The larger you got, the more competitive edge you had, the more you're able to beat up on the smaller cells and survive and not be consumed. In fact, a lot of eukaryotes were probably what was happening. Some of these bigger cells came around, they saw the smaller cell and said, oh, you look yummy. And so they ate that smaller cell. And they took it in. And it turns out that that smaller cell had a neat little trick called cellular respiration. And so we got our first mitochondria. And then some of these large cells continue to get bigger. The problem about getting bigger and only have to rely upon diffusion to get your energy, you only have so much surface area to allow material to move in. So one of the things you can do as you get larger is you can make your surface more convoluted. And so as you get bigger and then you have more space for diffusion to occur to get the energy that you need. And they believe that probably something like this happened, and eventually you had a DNA molecule here up on the membrane, kept getting bigger and bigger, and then basically it started making these folds, and one of these folds maybe something like this. It started to make folds around the DNA, so it got more surface area, And then we had to incorporate some more membranes here, and then brought these in, and so these started to grow out. And eventually they would fuse around this, and you end up with a nucleus. Which is again got that double envelope because of this, the way that it formed in the past. There was a way to deal with size, a way to deal with extra size to get extra surface area, and ultimately that resulted in a nucleus that allowed the cell to become more complex. And, getting bigger is better. It makes you more competitive and able to eat other things. And another way to get bigger and deal with size is to produce lots and lots of different cells and put them all together. Now you've got numbers. You've got more. More is better. I've got more cells than you, and so I can eat you. And some of these cells started doing specializations, like where they would actually make spines. They could paralyze their prey. And then they'd have specific cells that would allow them to digest better. So these would be rich in membranes for digestion. So we started seeing some of this first specialization of cells and tissues. And so multicellular life came into being, and multicellular life went from very simple to just a lot of cells and a nervous system and maybe some specialization to us and other organisms like us. So we believe, and the evidence suggests, and I'll show you in a minute here with some more evidence, that all of life to this day exists and is derived from a common ancestor and has adapted to the different environments on the planet. That's why life is so diverse. It represents not only an adaptation to a changing environment on the planet, but also historical contingency. This is things that happened over the course of 4 billion years has resulted in a lot of change. Multicellular life's only been around for the last, I want to say, about 600 million years. So it hasn't had as much time, but 650 years is a heck of a long time. Okay, you can see that. So along came a fellow named Charles Darwin, and he's thinking about this idea of descent from a common ancestor and the organisms adapting to different environments, and trying to understand and grapple with how did evolution occur? How did life change on this planet? We know it happened. No one was debating about whether evolution happened. No one debates that. Life has changed. The question is, how has it changed? That's where all the debate came down to. He went on a really famous voyage with the HMS Beagle, Her Majesty's ship Beagle, or His Majesty's ship Beagle, who was on the throne at the time from England. 1831, 1836, he went to South America, he went to Chile, Argentina, worked his way up the coast of the Galapagos Islands, and there he saw wonders. He saw finches who specialized in eating seeds, insects, cactuses. They'd seen finches that he'd never seen anywhere else on the planet, with specialization and adaptations for specific types of foods, in order so that they could eat those foods better than their competition and have enough offspring to carry on to the next generation. And it all derived from an ancestral finch. And he proposed, based on the work of Thomas Malthus and others, that these organisms have some of the ancestral finches on this island. There's all these diverse different types of ecosystems and food types to eat. And some of the finches were better eating seeds, some were better eating insects, some were better at feeding on cactus. And their descendants, there was a lot of variation in there, and some of those had better ability at eating seeds, insect, cactus, and those are the ones that reproduced in the next generation. They outcompeted all the other members of the population, and so their genes carried on. And then there's more variation because of mutation, and you ended up with more organisms that had specialized abilities, and the population of fishes became more and more specialized. So that now they look different, they don't breed at the same time. Organisms that specialize in eating seeds would only breed when they had enough seeds to have enough energy to breed. Those specialized in eating insects relied upon when the insect populations were high enough so they had enough energy to breed. So they had different breeding times, so they didn't breed amongst each other anymore. That's what they call genetic isolation. Their population became isolated. They didn't interbreed, so there would be even more, they call genetic drift. They changed more and more, and they became separate species. And you saw this for the finches, you saw this for the iguanas that were very specialized, different from the iguanas you see before, and the tortoises. And he came up with this idea that populations vary. And within a population, take population, all this variation, within a population there are individuals that are better adapted for a given environment than others. And they will get more food, they will have more energy, and they will have more children, and their children will carry their genes. And that next generation will even have more variations because mutations still happen, recombination occurs, independent assortment occurs, and there are individuals within that population who are going to be even better adapted to environment. They're going to get more food than others, and they're going to have children, and they're going to carry those genes forward. And slowly but surely, the population changes to become the best adapted it can for a given environment, and those who aren't don't survive or don't reproduce, which is the same thing. Their genes don't survive. So competition amongst the members of a population results in individuals that have the best traits surviving and reproducing, and that's what causes populations to change over time. A lot of times people think about evolution like this. They say, okay, you have, and there's a reason for it. You see short-term all the time. You have this organism A, and it evolves, and you get organism B, and you get organism C. But a lot of times evolution is more like A becomes B, and that becomes C. It's not always split and you get two different organisms. And this is the same organism, it's just changing, and although these will interbreed, this is derived from this. It's about a gradual change, and that's more about what we see on this planet than we are about two populations splitting between two species. But this has happened too. I don't want to make that sound too unimportant because populations have been geographically isolated. Continents have split, where one population is trapped on South America and another one's on Africa. Now they evolve separately under different conditions, and they become so different that if you bring them back together, they can't interbreed anymore. We've dabbled in a little evolution ourselves with artificial selection. Look at dogs. All dogs are derived from an ancestral population that look a lot like a wolf. Now we've got Chihuahuas and Great Danes, bulldogs. They're very different based on what people have selected. They've taken a group of dogs and said, I like that trait. So I'm going to let those dogs breed because they both have that trait. And all their puppies will have that trait. And the other dog they don't let breed. So whatever traits they have don't get passed on to the next generation. And then they look at the new generation and said, that trait looks even more like what I want. I'm going to select those dogs to breed. And that trait changes over time. Again, you don't get to say, well, now we can. Now we go with our DNA recombination techniques and maybe actually physically change the breed. But they didn't do that back then. They didn't have those techniques. All they could do is work with what nature gave them. They looked at the dog population, that these are the traits. What traits do we want? Let's breed for those traits. We do that over a long enough period of time. We see the dog population change. We get different breeds. We've done it with the foods that we eat. If you look at cabbage, you look at Brussels sprouts, you look at broccoli, they are all from the same genus. And they have been derived from our playing around with artificial selection. Selecting, like with broccoli, we're more interested in the flowers, versus Brussels sprouts, more interested in the leaves, basically the leaf buds that form off of the stems. There's a whole bunch of cruciferous that are all derived from this ancestral cruciferous that we worked with and made all these different foods that we eat to this day. And we've done that with wheat, we've done that with so many different crops, it's not even funny. There are other players who were also coming with ideas about how evolution occurred. Darwin is about, we don't create the variation happens randomly. He didn't even know how, he didn't even know about radioactivity and mutation and UV radiation. He just knew that in a population you always had variation. Others will look, and he said basically amongst those variants, only those that had the best features survived, and there were enough time that caused changes in the populations. Lamarck had a different idea. Lamarck said, he said basically, if you got a population of giraffes, and the food is getting a little too high for them, so they stretch their neck. And eventually they stretch their neck. So Lamarck thought this was possible. So by stretching their necks over their lifetime, they got longer necks, and they passed that on to the next generation. And the food got higher up because basically they were eating all the food that was low up, and then they stretch even farther. So they stretch their necks more, and then their next population, next generation has longer necks. So the idea was, it'd be like if you started going and working out at the gym, you got all real muscular, and you pass it on immediately to your children, the next child born must be equal. That doesn't happen. But the mark wasn't really that simple either. It was more complex, a way to create more nuanced. Nevertheless, it kind of was what he was talking about. Darwin said that the population of giraffes varied, that there were some giraffes with slightly longer necks than other giraffes. And the giraffes with longer necks got more food, had more offspring, and so the next generation had giraffes that had longer necks. He hasn't died. The reality is they just didn't get enough food to reproduce, and their genes that carried to the next generation. It's effectively the same thing. So these new giraffes have slightly longer necks. They vary. Mutation happens. You get a population of giraffes, some with shorter necks, some with longer necks. The ones with shorter necks don't do so well. The ones with longer necks get more food, and they reproduce more, and they pass it on to the next generation. But the variation occurs because of random mutation, random recombination. The environment selects the individuals that have the best variations, the best traits to survive. And that causes a gradual change. It doesn't take millions of years, it only takes thousands of years, and it causes the population to change. It doesn't have quite what they call gene pool frequencies, allele frequencies in a population. Don't forget Alfred Russel Wallace. Darwin sat on his idea for a while because he knew the implication. He knew that society was not going to be super happy with his ideas in regards to humans. And so he was bowling and writing. He was pretty kind of a downer guy. But he was brilliant and had great ideas. And his friends were always like, you got to publish. He put it off so long that someone else came up with the idea. He was out, this guy, Alfred Russel Wallace, was out in Tahiti and Fiji, and he was seeing all the Polynesians, and he was seeing the same sort of amazing variety of life out there, and ancestral species that were derived from new species that were derived from ancestral species. And he came with the same idea. In fact, he wrote to Darwin saying, hey, I have this idea, I'm thinking about publishing it, because he knew Darwin was like a famous field biologist. He'd been on the Chimpanzee Beagle. And he wanted his opinion, and Darwin was like, oh, what do I do? This guy's already come up with my idea. And his friends were like, publish now before he publishes, because you deserve credit, you came first. And he said, nah, he ended up sharing credit with the first paper on evolution by natural selection. But no one remembers this guy anymore. But Darwin went and wrote a book about us, the original species, and that's what started all the controversy. Alfred Russel Wallace was understood natural selection, but he would never really apply it to us. He said our intellect was derived from a different method. We'll say it that way. So let's talk about some evidence for evolution. Fossil evidence is probably the oldest evidence out there for evolution. So it's been around for the longest. It's what got Darwin and his contemporaries talking about evolution. These organisms lived in the past, didn't live anymore, but looked a lot like organisms that live now. So this is how fossils form. Basically, something dies, and then it gets buried. And then that's sedimentation. Sedimentation is the deposit of sediments of minerals on top of a previously existing layer. So organisms get trapped in them. The most famous fossil beds out there are believed to have occurred from things like mudslides, landslides, volcanic explosions that cause power casting flows. A lot of ash gets deposited on the organisms that die from the gases that are released by the volcanoes. So they die pretty instantaneously, they get buried pretty instantaneously, and then the next step can occur, mineralization. When you get large populations like that buried instantaneously, a lot of them will undergo this process. Not all of them, but a lot of them do. And the organic material, the living tissue, gets replaced by rock. But because it's building upon this model that was living, it takes the shape of what that living organism was. It's just replacing tissue, the shape remains. And later on, we come along, we start digging around in the ground, we start breaking open particularly sediments where you get shell-like rock, you start splitting open, and lo and behold, you see the bones of a fish. I'm thinking of a little fossil expedition I did a long time ago back in Wyoming. We did a lot of fossil fish. And they start, looking at it, these don't exist anymore. And they start making connections with organisms that did exist. So mineralization allows us to look at fossils. Paleontologists study it. They say the realm of paleontology is study fossils. And one of the big pieces of fossil evidence for evolution, what they call transitional fossils. This is looking at Tiktaalik. This fossil was predicted to exist before they ever found it. Neil Shubin, which will be seeing a video with Neil Shubin next week, I believe, was one of the scientists who proposed this idea. I don't remember who his collaborator was. And he basically was saying there's something out there that has some of these features of a fish and some of these features of amphibians. Because that was the big question. How did fish get out of the water and give us amphibians? And there's these bluefin fish that we believe was the most likely ancestor of amphibians. Because they're these fleshy fins that they could actually, and to this day, there are descendants and relatives of these that still exist that are still fish. They have the capacity to get out of water for short periods of time. There are other organisms like catfish, called walking catfish, and get out of the puddle and go from one puddle to another puddle, just so they also go find more food. So, what would be the advantage of a fish like this? I mean, again, get out of the puddle to find food that organisms, other fish can't get to because it's out of the water. Escape predators so they don't get eaten, and they can pass on their genes to the next generation. So a fish that was getting better at getting out of the water and staying out of the water, there was some definite natural selection that would go to work with it and maybe come up with something that looked more like an amphibian. In fact, there are fish that look a lot like amphibians, but they don't really have the ability to be on land for short periods of time. So there are some transitions, and you have to think. When we think of fish, we think of a lake, or think of a stream, or think of the ocean. But remember, there's marshes, there's places where there's very shallow waters, organisms that live among these reeds and have to have these fins that help them move and manipulate the reeds, get between the reeds to escape predators. So there's already a natural selection working even in these shallow waters to create the characteristics necessary to take the next step on the land. But there had to be some organism that had sort of an in-between, had some features like a fish, like a swimming tail, some lobe fins, but also had potentially some limbs that had the ability to actually crawl out. Unfortunately, we won't be watching the Shubin video that talks about Tiktaalik, although I think they show some picture of it. But he has like 3 videos, and if you want to, after you see that, you can go look it up on YouTube. They're available. I think it's a pretty interesting watch. And then, this is what they call transitional fossil, because it's between a fish and a full-blown amphibian. It has some fish features, it has some amphibian features, and probably live the life in between water and land. Although amphibians do that too. Amphibians typically only reproduce in water, and otherwise spend most of their time on land, although usually in a very humid environment. Another transitional fossil, Archaeopteryx here. We've known now for a while. In fact, if you think dinosaurs extinct, they're not. I mean, they are because true dinosaurs have certain features and birds have evolved beyond that, but birds are their descendants. Birds are the descendants of dinosaurs. A lot of features in dinosaurs, at least some of the dinosaurs is such a big category. There are so many different species and genera and family, and some of them are truly gone forever. But some of them survived, and their descendants are with us today as birds. Archaeopteryx is considered a transitional fossil because it had some of the features where it had feathers, right? It doesn't have feet, though. It's got teeth. It had claws that are very bird-like. With some dinosaurs never had claws that are very bird-like. That wasn't particularly unique. And now we know there are dinosaurs that had feathers. And they're still dinosaurs, they're not birds. But they had feathers probably to help keep them warm in cold environments. Somewhere along the line, there was a dinosaur that had feathers, and they were long enough to maybe allow it to glide first. And then eventually, through natural selection, to be able to fly and get places where other organisms can't eat you, and also get the food that other organisms can't get to, that's a huge benefit. And so even short flights could be beneficial for escape or getting to foods. So Archaeopteryx was one of these transitional fossils that was on the way to birds. Now birds are very different. Beaks. There were toothed birds that had beaks too. Another transition. And then, of course, this is a really famous example of the transitional fossils of whales. Ambulocetus is considered the ancestor of all whales. Was an organism that was pretty terrestrial. It had limbs, but it also tended to go a lot into the water to get its food, probably escape predators. It itself was a predator. It had a nose at the top of its nostril, which a lot of organisms do have that configuration. We don't. That was pretty good, but it wasn't as good as having nostrils saying even farther than the top of the head, so you didn't have to lift it as high to get your oxygen. So, Basilosaurus is considered a transitional fossil. I don't know, it's an ancestral well. But one interesting thing is to look at Ambulocetus, who actually has a full hind limb, and Basilosaurus doesn't have a hind limb or really much of even a fin there, but does have a residual pelvis complete with leg bones and feet bones. And even today's right whales, so the modern well, if you look in the region of the pelvis, you do find the residual bones of pelvis much depleted. And that's typically what we see in the organisms that where natural selection is not really acting upon something anymore at all. It doesn't use this anymore, so it's just kind of degrading over time. You look at their flicker zone, you still see the hand bones there. It's not like a fish's fin. So this is an animal that was once terrestrial, has come back to the ocean, and is now getting some of the same features as a fish has. And the nostril has moved all the way to the top of the head now, so it doesn't really have to lift its mouth out of the water to get its head. Biogeographical evidence for evolution. Biogeography is the study of the distribution of plants and animals. That's allowed us to see a couple of things. One is, like I was talking about earlier, South Africa and Africa were once one mass. They fit together like two pieces of a jigsaw puzzle. They were once one single landmass, and there are old world monkeys and there are new world monkeys. And they share similar ancestors, but they're very different because South America and Africa have split apart millions of years ago. There's what they call molecular clock we're talking about, where you actually look at mutations in a gene and you're able to determine how long ago two populations separated based on the number of mutations in a given gene. And if you compare that timeline, you look at that, what that data tells you about the separation for two different species, and you compare that to the timeline given to you by geography between when Africa and South America apart, you find those timelines agree, that the evidence supports each other. The idea that these species became separate because Africa and South America separated probably millions of years ago. The other thing about biogeographical evidence is they look at how organisms were affected by the geography of the region and the characteristics of that region, like hot and dry or if it's wet and humid. There are organisms that have adapted to these environments and this geography that's resulted in them looking a lot like each other, but they are derived from different ancestral lines. They call this convergent evolution. The Patagonian hare here is an organism that is derived from rodents, like rats and mice. And yet, it has very similar features to a rabbit, which is a lagarde, a completely different line. And because they evolved in an environment very similar to each other, they've adapted to some of the same features because the environment is acting upon their populations. They had some of the same variation, and yet the organisms look a lot like it. The euphorbias here are plants that look very much like cacti, but they're not. They're again derived from a different type of plant. Yet they have some single features. They don't have a lot of leaves. They have fleshy stems that have photosynthetic capacity. This allows them to minimize their water loss in a dry desert environment. They have thorns. They don't have cacti spines. The thorns are much more like the rose's thorns, whereas the cactus actually has much more pointed spines. But they still have that protective mechanism like a cactus has. So they, again, totally different organism from a different ancestral line. It looks similar to some cacti because it evolved in a similar type of environment. And the same story for the poor Tasmanian vulture is now extinct. This is a picture of the Australia, I think. And they actually call it the Tasmanian tiger too. But it was more canid, like more like a dog than it was any sort of feline. but it's not either. It's a marsupial. It had a pouch like a kangaroo. It had its offspring were really tiny and migrate from the pouch and feed from the nipples inside the pouch and eventually grow to a full-size organism that escaped the pouch. But it had very much dog-like features, whether hunted in fact or not, I don't remember. But the environment acted upon it just like it acted upon the dog population to create features that are similar to how it hunts serves an apex predator. Even though it's derived from a completely different line of mammals, the marsupials, whereas dogs are placental animals and do not have pouches. There's also anatomical evolution, or evidence for evolution, that I'm going to talk about here. One is homologous structures. The idea is that these organisms have a common ancestor. So unlike what we were just talking about, we're talking about organisms that have different ancestry. And these common ancestors would derive organisms that would make different uses of the same features, the same, in this case, bones. So different functions, same structures. And we're looking at comparing a whale, cat, horse, and a human. And if you look at the forelimbs of a whale, cat, horse, human, you can find all of the bones. One bone, one humerus or femur, leading to two bones, radius, ulna, tibia, fibula, too many bones, carpal tarsal bones, too many phalanges, particularly for a whale, where they elongate that fin. You find that pattern for all of these organisms. Even the horse has a remnant fibula that's formed part of its leg. Even though they use these for different things, they're all different used for locomotion. This is for swimming, this is for grasping, this is for running on hooves, more grasping here, clawing. They even have claws that are modified as you find with other organisms. So using the same building blocks, you come up with different variations. That's considered a homologous structure. We can see their relationship to each other based on having this number of bones. You don't see this when you look at fish, for example. They're different derivatives. Analogous structures look at convergent evolution. Basically looking at different ancestors now, but ultimately the structures were evolved to give the same function. So in this case, we're looking at the bat and bird. We'll talk about that in a moment. Now, both bats and birds are vertebrates, and they both therefore have limbed bones. And you can see some similarities in that you can see one bone, two bones, and finger bones and everything. But you can see that the birds have done nothing with their finger bones. They rely entirely on their wing flight or feathers. And bats, however, have grown their phalanges to support the structure of their skin to create a wing. So they're using very different structures in a way to create the same function of flying. So birds are relying on their feathers, bats are relying on the bones in their limbs to give them the support for their wings to create that flight. Fins, fish have rays that don't look like phalanges really at all. Whales have their hand phalanges along with the rest of their arm bones to create their fins. They both are able to swim, neither for the same function, but they derive from different structures from different ancestral lines. And so all this points basically is nature acting as a molding force on living populations and creating different forms and shapes in response to be able to get us nutrients to survive and reproduce. So vestigial structures like whale hips or appendix, which we have no use for anymore, removed surgically, our tailbone, the coccyx, we don't have a tail anymore, neither do our friends and relatives, but our monkeys do, and they're all derived from the same ancestral organism, but we don't use tails and monkeys do. So like I said, if you don't use it, you lose it. Even embryos, if you look at the embryos of different organisms, you see a lot of similarities. A lot of embryos have tails, including us, like other vertebrates. It's just our tailbones get absorbed. Most of them. There's a little bit of the coccyx stuff. And we all have pharyngeal pouches that can become gills or ears or maybe jaws or ear bones. We see our ancestral history to a certain extent by looking at embryos. So when you're comparing even a pig embryo to a chicken embryo, totally two different vertical lines, birds versus mammals, you still find pharyngeal pouches in both of them. You find with fish embryos too, these go on to be gills instead of jaws or neck bones. And you see a postnatal tail on both organisms. And so there's a whole set of genes called OX genes that are used by multi-cellular organisms that give rise to kind of the pattern of where limbs are supposed to be and how many limbs we're supposed to have. And people have done experiments with and changing those genes in things like mice and particularly flies. Have shown that limbs appear where there weren't supposed to be limbs before. So we know that we're built upon this sort of this architectural plan that's based upon this evolutionary history that we've heard from the past. It's worked in a particularly precise way to create what we are today. Now, I think I'll finish up here. So I'll see you guys all in lab. If you haven't turned in your paper, make sure you get that to me.